Hello and welcome. In this video, I'll introduce DiffModeler, an innovative tool for assembling large protein complex structures from cryo-EM maps with resolutions up to 15 angstroms. DiffModeler fits protein structure models into cryo-EM maps to assemble large protein complexes. But before fitting structure models, the map is refined using a recent deep learning technique called the diffusion model. It begins by emphasizing the backbone of proteins in cryo-EM maps using the diffusion model. Similar to how DAL-E generates an image given some text, this model generates a new refined map where the backbone density is more apparent given the original map. Next, AlphaFold-predicted structures, or any other structures, are fitted into the refined map using Vesper, our structure fitting program. Finally, DiffModeler assembles the full protein complex by combining the best fitting poses of each subunit using specialized algorithms. Thanks to this diffusion model, DiffModeler can assemble protein complexes via fitting at resolutions that most other methods can't handle. To demonstrate DiffModeler, I'm going to assemble a protein structure of EMD3340, which is a cryo-EM map of a 4-chain structure at 7.2 angstroms. So you will start by going to em.kiharalab.org, scroll down to the roadmap section, and find DiffModeler here. On the EM server, there are two variants of DiffModeler, one with template structure input and one with sequence input. For DiffModeler template, you will provide structure models in a zipped file along with a PDB configuration file listing the PDB file names along with your cryo-EM map in MRC format. For DiffModeler seq, you will need your cryo-EM map and just a FASTA file with your amino acid sequences. First, let's cover DiffModeler template input. This is the job submission page. The first thing you will do is upload your cryo-EM map. Then you will set the contour level. You can do this by clicking the Visualize with Set Contour Level button, and then adjusting these values until you can clearly see the protein density and none of the noise. For me, that's about 0.04. So I'm going to input this value in the contour level field. Then I'll set the resolution of my map, which is 7.2 in this case. After this, you will have to create a zip file of all of your single chain protein structures. Here I have my 3 protein structures. I'm simply going to compress them into a zip file. Then I'm going to upload the zip file. Next, you will have to write a config file which tells DiffModeler which chains which template structure belongs to. To do this, open up a blank file in a text editor of your choice. Start by writing your PDB file names in each line. Then, for each PDB file, you will specify which chains it belongs to. So I know that in my structure, there are going to be 2 copies of 1.pdb. So I'm going to specify A and B. There's going to be only 1 copy of 2.pdb and 3.pdb respectively. So I'm going to name this chain C, chain D. Save this as a text file, and then upload it in the website. Small note, for both DiffModeler variants, you have the option to do domain-based structure modeling. So if you choose yes for this option, the server is going to use sort2 to split each of the chains into their constituent domains, and then model the complex structure based on those domains rather than the whole template structure you provided. DiffModeler sequence is pretty similar. Select it in the roadmap section. Upload your cryo-EM map. Set the contour level just like before. Set the resolution. Then for DiffModeler sequence, you're going to have to upload a FASTA file of your protein sequence. Now, similar to the config file of DiffModeler template, you will have to specify which chains which sequence belongs to. So I know that this sequence corresponds to chain A and B in my final structure. So in the header for that sequence, I'm going to put A, B. Similarly, I know that there is one copy of this sequence and this sequence in my final structure, so I'm going to label the headers appropriately. After this, I will upload this FASTA file. Your sequences will show up in this box where you can check if everything's good. You can also make changes directly in this box if you want to. And then you will hit the upload button. Once the job is submitted, you will see this page. If you don't have an account, you should bookmark this page or save this job ID somewhere so you can come back to this later. But if you're signed in, it will show up in your job manager where you can access it at any time and check on the status. Once the job is done, head to Job Manager, My Jobs, and select the job. So the DiffModeler result is as follows. You will have a CIF file of your assembled protein structure. You can click this button to download the protein structure in CIF format, where you can visualize it in Pymol or Chimera X. You can hit this button to superimpose the map on top of your structure to see how well the built protein complex fits into the map. If you click this tab, you can also see how confident the AlphaFold-2 predictions are and how confident the fitting is. In conclusion, DiffModeler is a very powerful tool to assemble protein complexes from cryo-EM maps at resolutions other tools often struggle to. For more information, please check out our tutorial for more detailed instructions, or if you want to learn more about the tool, you can read our paper at Nature Methods. Thank you.